Good morning, dear friends. In the last class, we studied about the apparatus for photosynthesis. The photosynthetic apparatus include the pigments, the protein systems, the photosystems, and the electron transport systems required for the trapping of light energy. And we have seen in detail how the light energy is trapped by the photosystem 2 and photosystem 1, and how the cyclic and non-cyclic photophosphorylations occurs, and how the energy-rich compounds such as ATP and NADPH2 are produced. If you summarize it, we can say that there are two types of photophosphorylations: the cyclic photophosphorylation and non-cyclic photophosphorylation. In cyclic photophosphorylation, the pigment system 1, the photosystem 1 is get activated with light, and the electron passes through different systems like pyridoxine, plastoquinone, cytochrome complex, plastocyanin, and come back to the P700. During this cyclic photophosphorylation, two molecules of ATP are produced when the electron jumps from pyridoxine to plastoquinone and in the cytochrome complex. Whereas in the case of non-cyclic photophosphorylation, both the pigments systems are involved, where the first excitation of electron takes place in the photosystem 2, where the ejected electron passes through the plastoquinone, cytochrome complex, plastocyanin, and to the P700. The P700 system is also get excited with the photon. Electron from the photosystem 1 is transferred to FRS, pyridoxine, NADP reductase, and ultimately it reaches the NADP, where it is reduced to NADPH2. And the gap formed in the photosystem 1 is filled by the electron coming from photosystem 2. So it is non-cyclic photophosphorylation, where one ATP molecule is synthesized in the cytochrome complex, and one NADPH2 is produced by the enzyme NADP reductase. Thus NADPH2 and ATP are synthesized during light reaction, and these coenzymes are used up in the dark reaction. During non-cyclic photophosphorylation, ultimately the electron comes from the water molecule, where the splitting of water takes place, releasing protons, electrons, and oxygen. As we have studied earlier, it need not be water, it can be any donor of protons in such as H2A, where A can be O, oxygen in case of water, or sulfur in the case of H2S, where the purple sulfur bacteria and green sulfur bacteria, which are photosynthetic bacteria, utilize H2S as the hydrogen source and release sulfur instead of oxygen. So today, before going to the details of the dark reaction or Calvin cycle, we will see what happens during the photolysis of water, or why it is called as photolysis of water. It is nothing but the splitting of water molecule. We know that water is the most stable compound on the surface of Earth, as it is the highly oxidized product of hydrogen. But the energy from photons can split this molecule and release the electrons from water. That is why Rabinovich defined photosynthesis as the oxidation or photo-oxidation of water. But the recent studies show that rather than the direct impact of light on water, it is the pulling force from the photosystem 2 which splits water. A gap of electron is formed in the photosystem 2 due to the removal of one electron, and there is a deficiency of electron in the photosystem 2, and the photolysis of water is purely a chemical process where the water is split up into protons and electrons. It is a chemical reaction where the chemical compound is split by photons. Not only the light energy, but any energy-rich electromagnetic radiation can split water and release the electron. The water splitting reaction is catalyzed by the oxygen evolving complex of photosystem 2. The oxygen evolving complex is a protein-bound inorganic complex which contain 4 manganese ions, calcium and chloride ions. 2 water molecules are complexed by the manganese cluster, which then undergoes a series of 4 electron removals or oxidations, and these electrons move to the reaction center of the photosystem 2. Here we can see a water splitting complex or oxygen evolving complex. OEC is surrounded by 4 core proteins of photosystem 2 at the membrane lumen interface. Electrons extracted from water molecules along with 4 photons produce a singular O2 molecule. The molecular machinery for dissociating water requires the ability to store energy from the first 3 photons before the 4 provide sufficient energy for water oxidation. Then the OEC transfers 4 electrons one at a time to photosystem 2 via tyrosine residue in the reaction center. The system you have seen can exist in 5 oxidation states from S0 to S4. The OEC appears to have a core containing manganese and calcium. The exact mechanism of splitting of water is not yet understood, but we can expect a progress in the coming years, and it will have a high impact in the area of energy research. It will be a wonderful mechanism to trap the light energy directly. There are many hypotheses regarding the formation of this complex in the microbial system, where the manganese and calcium rich minerals in the early oceans and its incorporation into the protein complex systems in the membranes of the microorganisms. It is present only in photosynthetic plants and photosynthetic microorganisms, including cyanobacteria. Its mechanism of action is not yet understood clearly. Anyway, the splitting of water takes place there, and the electrons are transported to the photosystem 2, and the protons are released into the lumen of the thylakoid. A small paper by Umashankar Dolai regarding this oxidation states, and I have put it in the link is put in the description. You can read that article to have a view about the possible mechanisms of the action of this water splitting complex. So the ultimate source of electron in non-cyclic photophosphorylation is the water. The release of oxygen takes place from water molecule, and the end product of light reaction are the NADPH2 and ATP. The next step in photosynthesis is the reduction of carbon dioxide, which takes place during the dark reaction. So we will see how the dark reaction takes place in the next class. Thank you.